part two. Let's talk about gas exchange, how it happens, and how these gases travel through your blood. So, as we've been talking about, have these alveoli, the alveolus that allow for gas exchange, and that happens through diffusion, right? So we depend on something called bulk flow, which basically means you've got to have a mechanism to get the air from your nose into your lungs. And you also need movement of the blood from one place to the other, from one capillary bed to the other. But once we get into the alveoli and the capillary beds, or the capillary beds in your body, because they're over here, then we are dependent upon diffusion for the movement of gases actually through the membranes. But you got to get the gases there first, which is part one. So we've got the gases there. How does diffusion work? Well, you've already learned a little bit about diffusion. That's nice. So you'll remember that diffusion is the movement of our molecules from a high to a low concentration of gases, right? So our diffusion of oxygen, carbon dioxide is going to work the same way. We're going to move from high to a low concentration. And hopefully you also remember that these are small molecules that don't have a charge. And so they work through simple diffusion through your lungs into the blood, and from the blood out back into your tissues. No transporter proteins required, just simple diffusion. And something that's really important to recognize is the effect of distance on diffusion. So, for example, if you were to have a concentration of glucose in water and then measure how long it takes that glucose to diffuse through that water, that it just takes 1 second for it to go 0.1 millimeters, so just a fraction of a millimeter. And then it takes 3 hours for that to move just a centimeter, right? So that's not very far, but that's not going to sustain life, right? If you're waiting for diffusion for things to happen on a larger scale. So we don't. You want to see it in a graph? Okay. Let me get the right button. And so if we were to take that same data and graph it, notice that right? So as the distance increases, what happens to our diffusion rate? Decreases exponentially. Bam, right? So as the distance to diffuse increases, the rate exponentially decreases. Here's the good news. It's proportional to surface area, which means if we increase the surface area, we're going to increase our diffusion rates. And as you just saw, it's inversely proportional to distance. You increase the distance and our diffusion rates are going to decrease. So diffusion is only effective in our bodies on this microscopic scale. And that's what it uses it for, right? So let's talk about the structure of the alveoli and what helps aids it, how that structure aids this function of diffusion. First of all, you have a lot of them. You've got 300 million of these little sacs in your lungs, which are estimated to cover a surface area of 90 meters squared. So that's like a lot, right? Big surface area. Like that's way more than the surface area of your skin that you have inside of your lung. And it's really thin because diffusion only works in short distances. So it's about half a micrometer thick, super, super thin. And those are surrounded by capillary beds, right? So they're both lined with that simple squamous epithelium. So really short distance, lots of surface area for this diffusion to happen. So that in a healthy lung, if we look at as blood is flowing right from your pulmonary arteries into these capillary beds and then out through the pulmonary vein, the time they spend in that capillary bed, you'll notice is actually less than a second. So even though this is where your blood is the slowest, right? Remember your capillary beds, the speed of your blood is the slowest. It's still spending less than a second going through those capillary beds. But when is it fully oxygenated? yeah, about right here-ish, right? So takes just a little over a quarter of a second for in a healthy body for that oxygen to become full, or excuse me, for that blood to become fully oxygenated. And fully oxygenated blood has a, I know, yeah, more with physics. This will make more sense in a second, but partial pressure of 104 millimeters of mercury. Okay. So even if you're a bit sick, right, and the blood is in the capillaries for a longer period of time, maybe it looks more like this, but you might still get to full oxygenation. But of course, if you're really sick, then with even in that 0.75 seconds, the blood doesn't become fully oxygenated. Or you've lost a lot of surface area. As in, do you remember what disease we lose a lot of surface area of our little alveolar? Yeah, emphysema, right? So if the lungs have emphysema, you have a lot less surface area. So that's going to decrease your ability to get oxygen into that blood. Or if your lungs are filled with fluid, as in pneumonia or ARDS, then you have this really thick, you increase the thickness. And as we've just talked about, you increase the thickness. What happens to your diffusion rates? They plummet. And so in that case, 0.75 seconds may not be enough time to get the blood fully oxygenated. So there's some laws about this. We're going to stick with the big picture. So the air you are breathing is not 100% oxygen. It's more like 21% oxygen. Most of the air you're breathing in is actually nitrogen. So Dalton's law just says this, that each one of those gases that it's in the air, like nitrogen and oxygen and carbon dioxide, those each have a partial pressure that when you add them together, you get the total pressure of the system. So do you remember what 1 atmosphere pressure is on the planet at sea level? Yeah, 760 millimeters of mercury, right? Enough to push that column of mercury up 760 millimeters. So that pressure is a combination of the pressure from the nitrogen, the oxygen, the carbon dioxide, the methane, right? All of these gases that are in our atmosphere. So how do you calculate a partial pressure, you ask? Because just Jones and some dude doing some math. Which is great. I also love math. It's pretty simple. First of all, you need to know, well, what's the percent of the atmosphere of your gas? Well, let's do it for oxygen. So you'll notice for oxygen is 20.9. We're going to round that up to 21%. And then we need to know, well, what's our total atmospheric pressure? Well, it is 760. So we multiply those two things together, and then you get the partial pressure of oxygen in the atmosphere, which when you do that, what do you get? I hope I have this memorized, right? The number in my brain, I think, is 160-ish. I'm not going to do the calculations right now, but okay, fine. Push pause. I'm like double checking your work and finding out you were right. Yeah. So so the air you're breathing in, the partial pressure of the oxygen is 160 millimeters of mercury, presuming you're at sea level. Okay. Question. All right. Then let's move forward. So here's a fun game. Let's say you decide to go hiking up around Sonora Pass, and there's a sign there where it says 9,000 feet. At 9,000 feet, the total atmospheric pressure is lower, right? Because you've moved up, so there's less between you and the top of the atmosphere. So the total atmospheric pressure at 9,000 feet is 545 millimeters of mercury. So what's the partial pressure of oxygen there? How would you calculate it? And then why is it harder to breathe at higher elevations? Why do you have to, you walk a little bit and you're breathing much harder than you would if you were walking a little bit down here in the foothills? Guess what this is? Zoom room time. All right. Talk to you there. Okay, and the other one is Henry's law. And the big idea with Henry's law is this. Well, let me read it to you. Solubility of a gas in a liquid is proportional to the pressure of the gas near the liquid. Here's the big idea. Pressure gradients work like concentration gradients. That's all this means, is that if we have our alveoli and we have our blood and the partial pressure of oxygen is higher here and it's lower here, guess what direction our oxygen is going to diffuse in? Boop, that way. That's all Henry's law means. Great. So then you can look at an image like this and start to make sense of it. You can do this. We'll break it down. It turns out it's pretty simple. The first time I looked at this, I went, okay, it's too much information. So alveoli in your lungs. So as you breathe in that oxygen, in this air, the partial pressure of oxygen inside of your alveoli, 104 millimeters of mercury for oxygen, and it's around 40 for carbon dioxide. Now you will remember that it was 160 out here outside of your body, right? So why is it 104 in your lungs? Well, because of this. So if those red little dots represent the air, the molecules of air that are currently in your lungs, right? And so the red dots are the ones that's just currently in your lungs. And you start to breathe, and we're not going to track the new molecules. You'll notice that even after you breathe 16 times, some of the molecules that were originally in your lungs before you started this exercise are still in your lungs. So with every breath, remember you have that residual volume. You still have some air in your lungs. So you're constantly mixing the new air with what's in the old air. So that's why even though you're breathing in initially 160 millimeters of oxygen in that new breath, it's mixing with your old air in your lungs, which has a lower oxygen concentration. So in your alveoli, the partial pressure of oxygen is 104. Great. As that blood enters into the alveoli of your lungs, what is our oxygen concentration? Yeah, it is 40. So 104 is higher than 40. So oxygen is going to diffuse into the blood so that in your pulmonary veins, it gets up to around 100 millimeters of mercury, or pretty close to 104, right? And then notice, right, as it goes through the heart, right, gas exchange isn't happening, so it's still staying at 100 millimeters of mercury. And then it's going to get down into your capillaries. And now here's where our internal exchange is going to happen. And your tissues have a lower oxygen level. It's usually less than 40. And so now this blood is at 100. Your tissues are at 40. What direction is oxygen going to go? Yeah, Henry's law says it's going to go from high to low. So it's going to go through simple diffusion into your tissues. And then what happens to the oxygen? Yeah, it's used by your mitochondria, right? So your these tissues, as soon as they get it, they're going to use it to make ATP. So that's what's keeping the concentration or the pressure, the partial pressure of oxygen inside of your tissues lower than the blood that's coming in to them because they are using that oxygen in the mitochondria, right? So the concentration of oxygen is always going to be lower inside of your tissues than the blood that's coming in, unless your lungs aren't working. Okay. And then we can follow the whole thing back through, right? And then the same will be true of carbon dioxide, right? Where is carbon dioxide the highest in this diagram? Take a moment there. And does it make sense that that's where it's the highest? Yeah, it's greatest in your tissues where it's being produced, right? And then it's going to diffuse from your tissues into your blood, because in your blood, it is about 40 millimeters of mercury. It's more than 45 in those tissues, so it's going to diffuse, and it's going to get around 45 millimeters of mercury until it gets into your alveoli. And of course, your alveoli are around 40, right? So we're going to get rid of that carbon dioxide. So can you do this? This will be our Zoom room. Can you now think about ranking these from lowest to highest, or from highest to lowest partial pressure? What's on the oxygen side? What's on the carbon dioxide side? Okay. So diffusion rates, right? It's based on the partial pressures. It's based on surface area. It's based on distance. It's also based on solubility of our gases in a liquid. And carbon dioxide is way more soluble in water than oxygen. And temperature. Warm water holds less gas than cold water, right? If you're a fisherman, right? Fish like to hang out in the colder water because it has, in part, because it has more oxygen, which also explains this graph. When we look at the diffusing capacity of CO, which is carbon monoxide, and oxygen to carbon dioxide, right? Carbon dioxide is much more soluble in water, which is why it has greater diffusing capacity. So what is your body going to do about that? It's going to make red blood cells. So the reason why having red blood cells is adaptive and needed is because oxygen has such a poor solubility in water. So while you're carrying some of your oxygen around in your blood, just in the blood plasma, most of it's being carried around inside of these red blood cells because the oxygen binds and has a high affinity for the hemoglobin. And without that hemoglobin, without those red blood cells, even if your lungs are working great and fabulous and they have all the surface area that they should have, if you didn't have those red blood cells, the oxygen just wouldn't diffuse fast enough into that water and we'd still suffocate. So you need those red blood cells. Your red blood cells are made up of 4 polypeptide or 4 protein chains. Those are called the beta chains. And at the center of each one of those beta chains, you have this heme group. And the heme group at the very center of it has iron. And it's the iron that binds to the oxygen. So each one of your hemoglobin can bind for oxygen. Now, we had some fun with graphs in mastering A&P. Hemoglobin does something called cooperative binding. And what does that mean? Well, similar to enzymes, when the substrate binds the active site of the, let's do it this way. When, if this is my enzyme and I've got an active site and the substrate comes into that active site, the active site actually it changes shape, right? It changes conformation. It's gonna hold on to that a little more strongly. Hemoglobin is similar in that when oxygen binds to that hemoglobin, at first oxygen kind of has a bit of a hard time kind of getting in. And then, but once it does, then that heme changes shape and then the other hemes change shape, and they increase their affinity for oxygen. And that leads to something called cooperative binding and aids in both the loading and the unloading of oxygen. Okay. An example. Let's walk through this one. In your lungs, the percent oxygen saturation of hemoglobin, right? So remember that you don't just have one hemoglobin molecule, right? You've got thousands and thousands in each one of your red blood cells, and you have millions and millions of red blood cells, right? So even though one hemoglobin is either going to be at 0, 25%, 50%, 75%, or 100%, right? Depending if it has 1, 2, 3, or 0, 1, 2, 3, or 4 oxygen molecules bound to it. Because you have billions of hemoglobin molecules, we can get an average of all of those and a percentage. Okay, so in your lung capillary beds, oxygen has diffused into the lungs and into those red blood cells. And so in your lungs, you have 100% saturation of hemoglobin in those red blood cells. But if we look at the partial pressure of oxygen in your resting tissues, so imagine this is like your deltoid that you are resting and not using, then that deltoid is still producing ATP. It's still using that oxygen to make ATP. And so as that blood is traveling from your lungs, and it's going to go through the left side of your heart and then down and then out through your aorta and then through a series of blood vessels to your deltoid. Once that blood enters that capillary bed, what is the percent saturation of hemoglobin as that blood is entering that capillary bed? Yeah, it's still at 100%, right? No gas exchange is happening until we get to the capillary bed. And when it gets to that capillary bed, then this 100% here is going to interact with this 40 partial pressure in your tissues. And 100% is what? It's 100. It's kind of handy, right? It's 100 millimeters of mercury. And so, right, diffusion from high to low. And in this case, how much is going to diffuse out of the body? Well, this is how we read this. Well, at 40 millimeters of mercury, we're going to be right here. So that's around 75%, right? Ish. So what that means is about 25% of the oxygen that is traveling in your blood is going to unload and go into your resting tissues. Great. What if I start doing my best dance moves? Then I'm going to start using that deltoid, right? I'm going to start exercising it. So what's going to happen to my ATP production? Boop. What's going to happen to the amount of oxygen that's being converted to water? It's going to increase. So what's going to happen to my oxygen concentration or the partial pressure of oxygen in that exercising tissue? It's going to go down, right? So to like 20-ish or a little less than 20, 18-ish. So if I'm exercising, then what's going to happen? Well, now, as that 100%, 100 millimeter of mercury hemoglobin arrives into my capillary beds, well, now I've got a bigger concentration gradient between 100 and 20. So if I look at my graph, what's happening? Well, now I'm going from, it's arriving at 100% saturated, and that is dropping down to, whoa, stop, 25%. Holy moly. That's a big difference, right? From 100 to 70. Whoa. 75 versus 100 versus 20, that's a 25. And that's due to cooperative binding, that as those hemoglobins start to lose oxygen molecules, they're more prone to let them go. And as they gain one, they're more likely to bind. Basically, what this means for you is as you exercise, this curve is really steep in this area, which allows you to maintain most of your oxygen in your blood when you don't need much oxygen, when the oxygen demand isn't very high from your tissues. But as that oxygen demand increases, then you dump a lot more oxygen into your cells. All right. But wait, there's more. So normal body temperature is about 38 degrees Celsius. And what's the effect of temperature? And why would this be helpful to your body? All right, so where's that exercising thing again? Yeah, about right there. So here's my exercising tissue. Now, when I'm exercising, if I'm at normal body temperature, right, then I'm going to get about 75% of the oxygen unloading from the blood into my tissues, right? Because it's going from 100% down to 25%. But what happens when you start to work out, what happens to the temperature of that muscle? It's going to increase, right? Because you're using more ATP. So if my body temperature increases, check it out. Whoa, now I'm going to unload down to 10%. Now I'm going to unload 90% of that oxygen from the blood that's coming in. Is that handy? Yeah, right? Because if my body is warmer, that's an indication that I'm using more oxygen. And so oxygen unloads from hemoglobin faster. That's handy. If I get colder, what's probably going on with my metabolism? It's probably lower. Do I need less oxygen? Yeah. Am I unloading less oxygen? Yeah. So temperature affects how easily oxygen's loading and unloading onto hemoglobin. But what else, Erin? Well, you will notice some other things. So normal arterial carbon dioxide, right? Or hydrogen ions, pH of 7.4. If we increase the carbon dioxide, would you do that if you start to exercise? Yes. Or if our pH starts to go down or become more acidic. And you'll remember way back in part 1, right? You add carbon dioxide to water, you get carbonic acid. That carbonic acid dissociates and the hydrogen ion gets released and the pH goes down. So as you exercise, you would expect to see higher levels of carbon dioxide and lower pH in the blood. And you also see this shift to the right in that now we're unloading more oxygen from the hemoglobin to enter into those working tissues. So temperature, carbon dioxide, hydrogen ions, that all causes this shift to the right. As does a molecule called DPG. We're not going to get into detail on that, but that's also something that would increase as you are exercising. There's one more image. Nope, this way. All right, so that's oxygen transport. Most of it, a little bit is going to diffuse into the blood plasma. Most of it, though, because it has such a low solubility in water, is going to be transported in those hemoglobin molecules. And then it has this cooperative binding. And then those different factors are going to cause that shift to the right. So now let's talk about carbon dioxide transport. And this becomes important as your body is trying to regulate the pH of your blood. As you can see, carbon dioxide transport is a little more complicated, so we're going to walk through that. The thing I want you to notice on this slide at this moment is this, right? Oiled again, right here. That carbon dioxide is transported in 3 different ways. One, a little bit of it is just dissolved into the blood plasma. And then 23% of it is HGB-CO2. What does that mean? That is hemoglobin. So carbon dioxide also binds to the hemoglobin. So remember that shift that, that we saw in the unloading of oxygen from hemoglobin, that as carbon dioxide levels increase, then oxygen is not as tightly bound to hemoglobin and kind of jumps ship easier. That is because that carbon dioxide is also binding to the hemoglobin and causing it so the oxygen isn't binding to hemoglobin so strongly. And most of your carbon dioxide is moving through your blood as HCO3, which is bicarbonate. And we'll be talking about how all of that happens. Let's do it. So carbon dioxide transport. First of all, where are we in the body? Are we in the systemic circuit or are we in the pulmonary circuit? Well, we're in the systemic circuit because we're looking at tissue cells. So things like your liver or your back to the deltoid, right? So in this case, those red lines, what are you looking at? Well, that's right, oxygen, that's easy peasy. It's unloading from our hemoglobin and diffusing into the cells. And then a little tiny bit of it is just dissolved in the blood plasma, and that can also diffuse into our cells. So let's look at this carbon dioxide. So carbon dioxide is going to be diffusing out of the tissue cells into this interstitial fluid and then into the blood plasma. And some of it just stays dissolved in that blood plasma, right? About 7%. And then some of that is going to slowly, as we've talked about, it combines with water, becomes carbonic acid, and then carbonic acid is going to dissociate into bicarbonate and a hydrogen ion. What happens to that hydrogen ion? Well, remember, right? Ooh, pH. How are we going to deal with that? So you might remember when you were learning about blood that you have some proteins dissolved in that plasma that are helping to regulate the pH of the blood. Well, these hydrogen ions can bind to those proteins in the plasma, and that's going to help regulate our pH so it doesn't just tank, right? And it has some capabilities to do that. Now, the majority of that carbon dioxide is actually going to go into those red blood cells, and 2 things can happen to it. One, start with the smaller one. It can bind to hemoglobin. And well, I just reverse it if you want to go over that again. But it binds to the hemoglobin, and as it binds to the hemoglobin, that helps bump that oxygen off of hemoglobin. What happens to most of it, though? Well, most of it is going to, there is an enzyme in your red blood cells that is carbonic anhydrase, and it's going to bind as a substrate in its active site, carbon dioxide and water. And when it does that, it makes H2CO3, which is carbonic acid. And then that carbonic acid is going to dissociate into bicarbonate and to our hydrogen ion. And what happens? Well, if this builds up, then this whole thing, remember our equilibrium lab through your brain palace, right? And so when I do this, that means that our reaction can go in either direction. And that's true here, that this carbonic acid versus our bicarbonate and hydrogen ion, that can go in either direction. Do you remember what pushes it in one direction or the other? Yeah, the concentration on one side or the other. So if I get a lot of carbonic acid, which I would be doing in this capillary bed because I have so much carbon dioxide coming out of my tissues, then this is going to be the direction that this series of reactions is going to go. So I have lots of carbonic acid. So I'm largely going to be making a lot of bicarbonate and hydrogen ions. Now, as that starts to build up, then this is going to slow down. And then you can't carry away as much carbon dioxide. So here's what happens in your cells. That bicarbonate leaves the red blood cell, and that helps to continue to push this reaction towards making more, this whole thing can go back and forth, more carbonic acid, and I can carry away more carbon dioxide. Okay. But you'll notice that bicarbonate has a negative charge. So to maintain the charge difference so that there isn't one, there is this thing called the chloride shift, meaning that for every bicarbonate that comes out, one chloride ion comes in, and so then the charge remains the same inside and outside of the cell. Now, let's go back inside the cell. All right, so we explained this bicarbonate. What's happening to this hydrogen ion? Well, if we accumulate those hydrogen ions, what's going to happen to the pH of our cell? It's going to go down. That's going to be a problem. So hydrogen also can bind to hemoglobin. How about that? So some of your hemoglobin can bind to, so carbon dioxide can bind to hemoglobin, as does the hydrogen ion. And when the hydrogen ion is bound to the hemoglobin, then it's not going to affect the pH. Pretty cool, right? So that's how your body is mitigating changes in pH when it's loading carbon dioxide into the blood. That the hydrogen ions in the plasma are going to bind to these proteins, and the hydrogen ions inside of the red blood cells are going to bind to the hemoglobin. Okay, so now we're going to catch a ride through the vein, go to the right side of the heart, be pumped out through the artery into our pulmonary circuit. And now we are in the alveoli and the pulmonary capillary beds. And now the opposite thing is happening, right? So oxygen is being loaded into the blood and into hemoglobin. And as our oxygen is being loaded onto the hemoglobin, it's going to be that hemoglobin is going to be less attracted to that carbon dioxide. The carbon dioxide is also, right, it's higher here in the blood, it's lower here in the what is it? Alveoli. And so it's going to go from a higher to a lower concentration so that carbon dioxide comes off of the hemoglobin and to be exhaled. Same thing's happening with the carbon dioxide that's dissolved in the blood, right? It's just going to go from a higher to a lower concentration. Because this carbon dioxide is leaving, that pushes these reactions towards making more carbon dioxide. And same thing here, that as this carbon dioxide is being unloaded from the cells, diffusing into the alveoli, then it's going to take this equation that in your systemic circuit is being pushed towards making bicarbonate in your, in your alveoli. It's because the carbon dioxide is leaving the system, it's being pushed in this direction. And it's that simple. Nice. Let me know if you've got questions about that. Oh, and same thing here. Now, what do you notice about the chloride? Yeah, it's leaving. So as this, as carbon dioxide leaves, this carbonic anhydrase is now working in the reverse direction, and it's taking the carbonic acid and making more carbon dioxide and water. And so as this decreases, then I have lots of bicarbonate and hydrogen ions. And so the bicarbonate is also leaving, and that causes the bicarbonate to diffuse back into the red blood cell. And so then the chloride balances it out and moves outside of the cell. Very busy. Right, last topic, your breathing rate control. So let's talk about that feedback loop and where are the stimuli that affect your breathing rates? Where are those receptors? What's the control center? What are the effectors? All right, so breathing is really important. So it turns out you have lots of different inputs on what your breathing rate should be. I'll let you take a moment. There are many, right? Some of them you have voluntary control over in your brain. Some of them it's emotional, right? That panicky feeling or feeling really relaxed, right? Pain, different emotional stimuli are going to work through your hypothalamus and then talk to your respiratory centers in your medulla oblongata. Yeah. So your control for your breathing is in your medulla oblongata, similar to your heart rate. They're both being controlled in your medulla oblongata. And you also get some input from your lung stretching, or if you're breathing in irritants, or if you're starting to move your muscles around, that kind of sends signals to your brain. But the thing that is regulating your breathing rate for the most part, and most of the time, are these things. This is number one input, primary input, and that's your chemoreceptors. So remember, chemoreceptors, where else do you have chemoreceptors that you find to be delicious? It's going to pick up my beverage. It's tea, don't worry. Right, so you get those in your tongue, your nose, so they're detecting molecules in the body. But you also have chemoreceptors inside of your, well, where are those? You have two different types. You have peripheral chemoreceptors and central chemoreceptors. And I'll come back to that in a second. So your peripheral chemoreceptors are in, what do you notice? Similar place that your, those baroreceptors are. So you have them in your aortic arch, and you also have them in those carotid arteries in your neck. So you have these chemoreceptors that are monitoring chemicals in your blood. You also have them in your brainstem. And so these are your peripheral and your central. And now let's look at those again. So your peripheral chemoreceptors, those are monitoring oxygen concentrations, carbon dioxide levels, and those hydrogen ions. So all three of those things. And of those three, it's mostly paying attention to carbon dioxide and hydrogen. Oxygen doesn't really have a thing to say unless it gets really pretty low. So peripheral chemoreceptors, oxygen, carbon dioxide, and hydrogen, but the two most important, carbon dioxide and hydrogen ions. In your brain, in your chemoreceptors, you'll notice your brain actually isn't keeping track of oxygen levels. It's concerned with changing the pH because that would be really damaging to your brain. So it is monitoring your carbon dioxide and your hydrogen ions. And really specifically, these hydrogen ions is what it's looking at mostly. That as that carbon dioxide binds to water and produces carbonic acid, then it's detecting that change in pH to do those hydrogen ions. And that's happening inside of your medulla oblongata. All right. So let's walk through this one. So if I have high arterial heart partial pressure of carbon dioxide. So your carbon dioxide levels in your blood are high. That's what that means. What are we going to do about it? Well, let's follow this side first. So your peripheral chemoreceptors, which are in the aortic arch, as well as your carotids, right? They are going to mediate, on average, around 30% of your response, right? So remember that they are monitoring carbon dioxide along with the hydrogen ions that are increasing due to the increasing levels of carbon dioxide in your blood. And that's about 30% of your central control's response of what your breathing rate should be, how many breaths per minute. The majority of it, though, is happening in your central chemoreceptors in your medulla oblongata. And that increase in carbon dioxide is going to cause a decrease in pH, right, or an increase in hydrogen ions. And that's going to trigger or stimulate the central chemoreceptors. So as I said, right, these are mostly responding to those hydrogen ions as a measurement of how much carbon dioxide is in your blood. So that's the primary thing that's telling your body to breathe, is this buildup of hydrogen ions. And about 70% of your response to how quickly should you be breathing is happening from input from those central chemoreceptors in your medulla oblongata. And then in that medulla oblongata, you have your respiratory center, and that is going to talk to your diaphragm and your external intercostals. And if you need to, well, what do I need to do? If I'm got a high carbon dioxide levels, do I need to increase my breathing rate or decrease it? Yeah, I better increase it, right? If I increase it, then I'm going to exhale more and get rid of that carbon dioxide. So I'm going to send an impulse to those muscles to contract, right? Because when those contract, that's when I inhale. And those pulses are going to be sent more often, so I am breathing more frequently. And then my arterial carbon dioxide and pH levels should return to normal. And carbon dioxide and hydrogen levels tend to change more quickly compared to oxygen levels. So by monitoring the carbon dioxide and hydrogen ions, if that's taken care of, then one, you're taking care of the blood pH, and two, if that is properly ventilated, then if all is well with your system, then that will also properly oxygenate the blood. And there it is in more detail. I'm not going to ask you to break down the respiratory centers, but there they are. Okay. Thanks for sticking with it. I look forward to seeing you in the Zoom room. Or if you've got questions, I've got office hours, I've got email, you know how to find me. And you've also got some great tutors. So hope you are getting the resources you need to feel like you are learning. Talk to you later.